be there. So when did you come in? Where did you come from?
Sure. I've been here since January 2010, so 7 full years at this point. I had the pleasure of working with Dr. Shwankowski and Dr. Grama, and every minute of it, I appreciate the time and support that they have had and they have, helping me in every step of it. I got my master's in December 2012 and been working on a whole bunch of different problems. So the ones that I'm going to talk about basically are the work of past couple of years, the major projects that fall within the same scope. But before that, we worked on a whole bunch of different problems, from network alignment with Dr. Gulaik and aging for 2 years. For some reason, I thought aging is something that a CS student should work on. But so yes, there was a whole bunch of different problems, and these 3 projects that I'm going to talk about are focused on looking at cell type specific analysis and tissue specific analysis over different data sets in biology. So far, we had this generic analysis of interactions between proteins and activity of genes and activity of proteins, but never had context. So interactions where, in which context, in which tissue, in which cell, during which disease. And what we were trying to do is to add that context to this whole analysis. So we are adding one additional dimension to say interactions between proteins in this given context in this time point. So we're adding dimensions to analysis that we had previously, and that's the main contributions that I'm going to talk about from different problems that are related to this basic scheme. So that would be the main introduction, and I have one big picture that I like it. I spent a bunch of time on it. So I'm going to talk about 3 main projects that I worked on past couple of years, as I mentioned, and all of them start from the same point. We have a mixture of cells or cell types where they can be tumor biopsy, they can be blood cells, and they can be a bunch of different cells that are mixed coming from different tissues. And what we want to do is to analyze them. The first problem that I'm going to talk about is about a fascinating new technology that allows us to look at every single cell collected from a biological sample under a huge microscope at high resolution and with very high rates. One of the problems that we are interested in solving here is that we thought we know what are the cell types. Now we understand that there's huge diversity among things that look the same, but functionally are very distinct from each other. So one of the interesting problems that we try to solve is given that these cells are scattered in a high dimensional functional space, can we identify groups of cells that behave similarly, that have similar characteristics? In other words, can we identify these de novo cell types and subclasses? That will be part 1 of what I'm going to talk about. Given these cells, can we identify these groups that have similar behaviors? Then I'm going to look at the other side of the coin, and I'm going to say, let's start with a mixture of cell types and ask the question, can we separate these cell types from each other and look at individual components and their relative fractions? In an analogy, let's say we have a smoothie, a tasty smoothie. Can we separate the taste of banana and strawberry from it and say how much of it is in the mixture? This problem is also known as blind source separation among signal processing community. And then I'm going to change gear and look inside a cell and say, we knew that these proteins are supposedly interacting at some point in some spatial context. Can we say which one of them do actually interact in one specific time frame given one specific cell? Can we identify these cell type specific networks and cell type specific interactions? So to go over each one of them, I will separate into methods and results, of course. And the first part is a project that we've been working on recently, hoping to send it to Nature Methods, we'll see. And this project is a method for cell type identification, but also not only identify cell types, it looks at the problem of what makes a cell type a cell type. Can we identify key regulatory elements that make one cell type different than the other? So not only we want to do some type of clustering or grouping of these cells, but also we want to have interpretable reason as to why this cluster or this group of cells are different than the rest of them. So this method consists of 5 major steps that fall into 3 components. I'm going to over each one of the steps individually and then conclude with results. So the first step is that we try to define a proper metric space to represent functional relationship between cells. To give an overview of what the data looks like to begin with, we have a big matrix of genes by cells. So for each one of these cells, we have a high-dimensional signature vector that says the quantity of each gene, which has around 20,000 genes. And we have around, at the moment, around thousands of cells that we are going to look at. And in this space, the problem is traditional methods to identify distance don't work at least quite well. So if we want to use Euclidean distance or other measures, we have multiple problems that I'm going to talk about. Number one is that these signature vectors are dominated by these ubiquitously expressed genes that are expressed everywhere at high levels. So we have a lot of non-zeros that are useless for us, but they influence the distance between cells significantly. For example, we have genes related to cell cycle, which goes around and goes around and goes around, but in reality it doesn't have anything to do with the identity of cell. Also, we called out the noise portion of it. So we want to remove that or reduce that noise portion. And on the contrary, on the other side, we have these genes that are really critical for identifying the identity of cell, but these are only expressed at very low levels. So what we want to do then is to boost the contribution from these weakly expressed genes to enhance the identification of similarity between cells that we want to have.
How do you know that you're in noise, not signal?
Well, I define noise as what is not informative regarding the identity of cell. So if it's, I'm going to talk about how we compute this second portion. We use entropy and say one gene is useless or we don't care about if it's uniformly expressed across cells. If it's expressed almost nowhere, but in very limited context is very high, that gives us a lot of information regarding what that cell is. So the first part is to say what is the common factor between these cells and remove that common factor. So we know these genes are expressed at very high value but are uninformative, so we want to remove them from the context. But we need to know how does it look like, and we need to represent that matrix as a low rank approximation. And there are multiple choices that we can use, SVD or NMU, which is SVD with positivity constraint. And if we want to add interpretability, what we can do also is to add sparsity, to say don't remove everything, just remove factors that you know we have to remove. After we find this low rank representation of housekeeping genes, which I represent with a vector that represents, this is the dimension of housekeeping variation. What we want to do is that this is one cell type, this is the other cell. What we want to say is that I know this housekeeping exists, I know this is part of your identity, remove that part and tell me what else can you tell me. So we projected it orthogonal subspace of that housekeeping genes and ask, so how can we identify distances or similarities in this adjusted space? So the rest of it is just identifying that housekeeping signature, projecting everything onto that space. So we talked about the first portion of it. The second portion is how do we enhance or boost the signal coming from this this cell type specific genes, and we don't have any knowledge of which genes are actually cell type specific. So we need a method that tells us how cell type specific one gene is. And in order to do that, we say, let's say we have n copies of one specific gene, and we have 3 cells. N1 is in one cell, N2 in second, and N3 in third. We define probabilities as N1 over N, N2 over N, and 3 over N, and basically define a categorical distribution and say, what is the entropy of this distribution? How much information does it give us knowing that this gene is coming given the signature of that gene, how much information can be gained about the cell that it comes from? Any question regarding that part? Or can I? Okay. That was easy. So then this metric that we define induces a topology over the functional space of cells. And what we need to do is to basically identify this convex topology, which is assuming that there exists some cells that are pure cells that perform one specific function, and the rest of cells can be interpreted as combination of these pure functions that are performed by cells. Can we fit a compact representation over this space that tells us what are the functions of the rest of cells? And we want a compact representation, so we don't want to do overfitting, and we don't want to like fit to noise, because we are using this convex hull, which is fairly sensitive to outliers, but we want to ignore them. We want to have as many data points as we can fit in this red portion without with limited number of coordinates that, like k, we are going to give as a fixed parameter for now. And in order to do that, we start by saying, let's find exact k cells. We have these cells. Pick k cells that are maximally distant from each other in a sense. So basically what we want is to find k points. So H matrix in both of them is a column stochastic matrix. That is to say, each one of these cells, we want to interpret it, we want to write it as a convex combination of the other ones. The ones that fit in between are going to have 0 error. The ones that fall outside of this is going to add to the error. So we want to minimize that error first. And then what we want to do is to shift this representation and allow contributions from the rest of points. So initially we only allow each corner to be exact cell points, and then we allow other points. So here, instead of exact points, we allow contributions from all points. So it's a relaxation of the first one. Okay, so far I assume that I know k, but in reality I don't know how many cell types do I have, how many coordinates do I have to use. So I fit different values of k from 2 to a maximum k, which is 50, the ones that we tried. And ask the question, how much of these data points can I fit? And I look at that curve and I observe that it has an L-shaped curve. So I want to find the point with the sharpest turn and say at that point I have almost as much of the variance as I want explained. And after that, it's just overfitting to the data by adding these corners to maximally get the points. So I need somewhere here to say this is the point I want. And how I do find this is by fitting piecewise linear models. And I use 3-piece because I want to distinguish between rapidly shifting and slowly shifting curves. And then I use either the first breakpoint or second breakpoint. So if it's a rapid shift, it goes up, and there are 2 pieces that are basically same slope. And if it's slow shifting like this one, the second and third segments are going to have significantly different slopes. Finally, we have this representation of these archetypes of corners, and the question is, what makes a corner uniquely different than the rest of corners? And in order to do that, I say I have k corners, let's say 3. I look at this single corner and I want to say what defines its identity. So I orthogonalize it with respect to everything, every other archetype minus the one that I'm looking at. I say, tell me what do you have that no one else has, and that gives me a residual expression that tells me these are the things that I have that others don't have. Then I rank every gene according to this residual expression and ask, if I look at one transcription factor, which is regulatory element that defines the cell type identity, and look at how many of its targets are in the top ranked portion of this residual expression, that gives me a sense of how important targets of that transcription factor are for identifying the identity of one specific cell type. So first portion just talks about that, finding residual expression. Second portion talks about assigning a p-value to targets of a transcription factor that is among top ranked targets of a transcription factor that are among top ranked residual genes. So all that is amazing and good. How does it perform? We looked at 3 different data sets that are published in the past couple of years. So this technology started this dropstick, for example, the technology came out 2015. And so all these cells, number of cells that we can monitor is rapidly growing. Currently we are in order of thousands of cells, but the person that actually developed this technology, I've been talking to her, and she is currently working in 50,000 cells. And so basically, previously we were looking at the tall skinny matrix. Now we are looking at almost a square and getting even beyond that point. So the type of algorithms that we use completely is going to change in that regime. So we looked at 4 different data sets that come from different technology, some of them from older technology, some of them from single-cell RNA-seq, some of them from flow cytometry, and different number of cells that we are going to look at. And the first one is, how well does this metric that we define work? So we looked at this metric and compared it with one other measure that was specifically designed for single-cell data set.
I'm sorry.
Sure.
What's the final metric?
Final metric is, nope, this guy. Similarity between cells. So we have had this framework that we started from this matrix, and then we removed housekeeping, we boosted this similarity between, I forgot to say this is the one that we finally use. So Z orthogonal is after removing the effect of housekeeping. And then this is the weight factor for tissue specificity of that gene. And the final kernel that we define is that orthogonal weighted by tissue, like cell type specificity by this transfer. So it's a weighted dot product between these vectors that we are looking at. So we use this one and say, let's look at, like, do a one-to-one comparison, line up some different measures of similarity between cells or some kernels. One is the one that we defined, one is similar where Serafin Batul at Stanford developed, and 2 of them are general purpose dimensional reduction techniques. And we say, if we use these techniques and define similarity between cells, can we use kernel k-means to, given the known number of cells, to identify cell types? How well do they perform for the ones that we know what are the cell types to recover that? And what it shows is that these are for each data set and these are for each method. So what it shows is that in every data, and these are 2 different measures, one is normalized mutual information, the other is adjusted RAND index, which are 2 measures of quality of clustering basically. What it shows is that over each data set, over both measures, we outperform others in terms of quality of clustering or identifying cell types. So here we are better, and better. Same with both measures. So far so good. We have a measure that tells us how similar these cells are. Can we use this and combine it with that geometric approach that we just talked about to have a good quality algorithm that identifies cell types? This is just with known number of cells and with traditional kernel k-means. We can do much better than this. And then we compared it with state-of-art methods that are designed for cell type identification with different techniques. And we said, how well do we perform if we compare one-to-one using, again, same measures, NMI, ARI, and with 4 different additional methods, and see how well do we perform. In all cases, we perform better, except with melanoma data set in one measure, which what we found later is that we evaluated using the annotations that was given in the data set. What we found are things in more details that was given in the data set, and we validated and showed that what we find is actually true cell types that was not known beforehand, and that's the gray portion on top. If we add those annotations to the data set, how much does it change the validation that we are going to have? Finally, we say this is good, we found these cell types, but this is a continuous space. How does this space look like? So we use this projection onto 2D, which we start from Fiedler vector, and then we modify it using this nonlinear dimensional reduction technique called Ph.D. To project every data points in a 2-dimensional space. And then we mark each archetype using a unique color. We color every cell using convex combination of colors in these archetypes. So what it shows is that some cell types exhibit a continuum-like behavior. So here we are looking at lymphocytes, specifically T cells, and what we observe is that T cells are actually not distinct from each other, but they have very continuous transition between these states. Whereas a lot of cancer cells behave like distinctly different from each other. Same with macrophages and same with endothelial cells. But one interesting factor that we found is that two specific archetypes in this space basically form an axis. From A5 to A10, what we did is that we defined a line like axis between those two, projected all those cells onto that axis, and measured this MIT-F is one of the well-known markers for melanoma patients, and these are all cancerous, though these are all melanoma tumors. What we observed is that as you go from A5 to A10, the level of that MIT-F almost linearly decreases. So in a sense, we found an axis that meant something. The question was, what does it mean? Then we constructed that network.
Can I go back to the previous slide?
Sure.
So what is happening in these CD4 and CD8 T cells? Why
CD4,
archetype A1 and A2?
A1 and A2, okay.
So why do you see such a continuum on these CD4 and CD8 T cells?
So what happens is that technically, if we look at it, is B cell, T cell, then T cell distinguishes to two separate groups. So what it says is that the parent cell for NK looks more like CD4 T cell in that higher order differentiation tree. For some reason, these are just markers, known markers for CD4 T cell. It might be some intermediate cell that we don't know. These are all marked with what we already know about differentiation process. These are marked with limited number, let's say tens of markers.
See a continuum between CD4 and CD8 T cells. Why do you see a continuum
Well,
between CD4 and CD8 T cells?
I don't necessarily agree with that, because transition between cells might be due to interactions between environment. So what we see is that these T cells are within the blood cell, and based on the signaling between those cells, they decide to transform in or differentiate into one group versus the other. And that transition or change between the stages is a continuum by itself. But this whole area is fairly new. So before we thought these stable states of cell types is, I know this is CD8, this is CD4, and these are stable states in the dynamical system that we are modeling. And if you want to shift, there is a transient state, and then they are going to stay.
Take a cell that is between those two.
This guy?
No, take a cell between archetype A1 and A2. Take one of those blue cells.
Okay.
Is that a CD4 cell? Is it a CD8 cell?
This guy?
Is it a precursor cell? What is it?
We don't know.
What is it tagged as in the data set?
We don't have archetype here. We only have archetypes there, and I can look at the signature over residual vectors of the archetypes, not intermediate states so far. So the technique that we developed looks at residual expression of the hypothetical cell types or archetypes that we found, not the intermediate stages. Yes, what we can do is to then define a window here, but that's a hack because this is just a projection over 2D. It doesn't mean that in the high dimensional space they're actually the same topology. This might be an artifact of the technique that we use to project it into 2D. So finally what we do is that we look at the network, underlying network, that distinguishes this axis from the rest of the tumor cells. And interestingly, automatically we identify MIT-F as one of the factors. MIT-F is almost the only known transcription factor that they use as a biomarker to identify metastatic status of these melanoma patients. They say if this is expressed at high level, that's a better tumor. There is less chance of metastasis, whereas it's low, it's going to be a bad prognosis. We automatically found this for this subgroup to be associated in MIT-F, and also interestingly, other factors that are downstream of MIT-F, the ones that are marked as black are the ones that are known in the literature and known experimentally as biomarkers for this melanoma subgroup. And what we observe is that there are multiple functionally distinct regions there are 6 transcription factors out of more than 1,500 that we found to be relevant in this condition. And for each one of them, we looked at reasoning behind them and found like evidence in literature that shows evidence in different types of cancer that they have been known to be related, or sometimes we found experimental evidence in actual melanoma patient that this marker is relevant to prognosis of melanoma patient. So this network allows us to suggest some new avenues to say these are markers, these are therapeutic targets that might be of interest to understand this melanoma stage better.
And the network comes from what?
We are building this network. So the method that I developed, the method that I told you about, looking at the residual expression. So we looked at each archetype, specifically here at the MIT-F associated archetypes. We computed residual expression for genes in that case. We ranked all genes according to the residual expression with respect to MIT-F associated archetype. Then we looked at each transcription factor and asked how many of your targets are top ranked. If a significant portion of the targets, so these are for each one of these guys, there were more than 1,500. For each single one of them, we looked at targets that are high ranked in the residual expression profile. If there are a lot of them that are high ranked, or in other words, they have high residual value that is not explained anywhere else, that transcription factor has a significance in terms of identifying something about identity of that archetype. Combined with the targets that fall within that region. So we look at different cuts, and we look at the cut that gives us the maximum p-value after correction for multiple hypothesis testing, and say targets, so this guy, this transcription factor, has a lot of targets, some of them in top rank, some of them bottom rank. So what we say is that the cut that gives us the best p-value together with the targets that are in that top portion is the network we are constructing. We say this transcription factor together with its high residual targets are part of this network. So we found 6 transcription factors to be significantly associated with this phenotype that we were looking at.
Just ask one quick follow-up. Where did the original data on the transcription factors come from?
Okay, so there is this network. So we start from a transcription and regulatory network, a generic, so these are all possible interactions between transcription factors and target genes that can happen in any given context. Some of them might be false positive, some of them might be missing. So what we started is this network where it's a bipartite, transcription factors versus targets, and what we said is that which portion of this network of big, by let's say more than 1,500 by 20K network, which portion or which subset is related to the phenotype that we are looking at or we are interested in. And that's the initial data together with residual expression that we are looking at.
Shane,
Yes.
those hubs that you have, those are known hubs? Or
Which
those
ones?
different- So
These
the
guys?
ones in yellow.
This are not known, this is not known. MIT-F is the only one that is known. MIAX2 is actually known for the opposite. So we have 2 complementary subtypes, proliferative and
Come up with those hubs through your analysis?
okay, yes. All the hubs are from the analysis directly, and MIT-F is the only well-known one, and MIAX2 is also known how it behaves in melanoma patient. The rest of them are not known to be related to melanoma, but there is evidence, experimental evidence, that supports the fact that these are factors that should be relevant to melanoma. Okay, so then we are gonna look at the other side of the coin and look at the convolution problem. Now I have a mixture of cell types, and I want to separate them from each other and find how much of each one do I have in the mixture. To give you an example, why do I care? One of the best well-known example is that in tumor microenvironment or surrounding of this tumor, there are multiple subtypes of cancer. Even when we say melanoma, when we look at the biopsy, some portion might be resistant to some therapy, some might be responding well. So there are multiple subcolonies of these tumor cells. But also more importantly, there are additional cells that are hijacked by cancerous cells to perform some functions. And some of them will have significant importance, clinical importance. For example, these tumor-infiltrating lymphocytes have significant importance in immunotherapy and how well does it behave. What we want to understand is how does the surrounding of this tumor microenvironment look like? And in order to do that, we formalize it as a matrix decomposition problem where we have this matrix M, which is genes by sample. So each sample, for example, is a tumor biopsy. And each sample is a mixture of all these cell types. So I want to decompose it as GC, where G is the same genes, but their value in different cell types, purified cell types. And C, so this one tells me how does each cell type look like. This one tells me how much of each cell type do I have in each sample. So what I want to find is a good GC decomposition that mimics the behavior of M or characteristics of M. So if I want to model that, I want to define a distance function between that GC product and M, that distance might be Frobenius norm or some definition of the distance. And there are some restrictions on GNC. For example, we don't want fractions to be negative. Also, hopefully we want them to be sum to 1 so we can interpret them as fractions or percentages. So there are some restrictions on GNC. So there's a feasible region there, and we want to look at that region and find a good GNC that satisfies that property. So
When you believe this is an additive process?
so they
One cell next to another cell, you're assuming that the transcriptomic profile of these two cells together
it's just the additive of
is some scaled addition of these.
so they showed that this work.
But you know that there's all kinds of intercellular signaling that happens. You
Sure.
know that putting one cell next to the other will change the profiles of both cells
Completely agree.
in a very non-linear way.
Sure. What they showed is that on average sense, you can mimic the behavior as a linear instantaneous signal mixture. And what you can say is that yes, there are some effects, but on average you can mimic that as a just additive process.
But you're ignoring all of the intercellular signaling.
Yes. We can't measure intercellular signaling so far, which I'm really interested, as you know, to measure it, but we can't measure it so far.
You can measure it, but it's another thing to not even model it, right?
Yes. What you can measure, you can't really model how does it affect as a feedback loop to the system, right? So
You can use a non-linear mixture as opposed to a linear mixture model, and then you don't necessarily have your beautiful matrices and multiplications, but the combination.
some function of them, like f of this guy versus the other one. Might be, so what you're saying is that this G itself might not be similar to the purified one, which is actually true. In reality, what we say is that if you get references from somewhere else, it's going to look different than references in your own profile. That's one of the problems we want to solve. That is to say, given my mixture, how different this one is, assuming that the population has a majority behavior. So all the cells behave within, so the majority of them are inside that environment and the outer ones are in the minority, then you can model that whole microenvironment using a modified version of cell type. So you say T cell from somewhere else comes here, then we modify it to match with the T cell in our environment. So different methods were proposed in the literature, and people were mix and matching these features, different loss functions, different regularizers, different pre-processing, feature selection, all these different features, and they were claiming to be better. And the question was why they are better, which feature in their formulation made them look better, and can we mix and match those features to have an optimal configuration that provides some guarantee in terms of the behavior over different data sets. So we looked at different combination of them. So we started from loss function and said different people are using different form for solving that. Some people use the traditional L2 norm, which is the most common one in the square type fitting. And some want more robust form. They go with L1, which is not as sensitive for small values of error, but it's good for larger ones. It's less sensitive in larger ones, so it's more robust. And then Huber came out and mixes these two properties for small values as behaves like L2 for larger values as L1, so it's both robust on large noises and also sensitive in small. And then there's also this method recently came out, CyberSort, that people were all excited about. It was published in Nature. It just used SVR out of the box, applied it to this problem, and said, we are so good that we beat everyone. The question was, is it better because of regularizer? It has L2 regularizer. Is it because of loss function that has epsilon insensitive loss function, where it says if error is less than this, just ignore it. We don't care about like less than this. Within that region, whatever happens, no one cares. If it's outside that epsilon region, then start penalizing it with some linear fashion. So just to visualize it, this would be epsilon insensitive loss, where within one region, just totally ignore it. After that, linearly penalize it. This would be L1, this would be L2, and this would be Huber, where it behaves L1-ish, and then switch to L2, then becomes L1-ish. So we wanted to ask which one of these loss functions behave well, and which one of regularizers for shrinkage versus, so shrinkage is used in SVR, this one is more in loss of formulation, and what we wanted to do also is to say how pre-selection of genes affect the deconvolution problem. So what we are interested in is to find, so this is a highly overdetermined problem. What we want is to say, I know all these genes might be related, but which one of them gives me better conditioning over the matrix that I'm looking at? In a sense, I want to orthogonalize the matrix that I want to solve. But also, I want to find genes that if I look at them during different times, they behave similarly. Because one cell type that I have referenced might be in different condition than what I am here. So I don't want genes that look cyclic or time dependent, or I don't want genes that are flat pick. So the second one are signaling proteins or genes that respond to microenvironment. So they see something, they have a peak, they drop down. They see something else, peak, drop down. I don't want those type of genes because they are not consistent across time, or the time-dependent genes that are also dependent on time. I want invariant cell type specific genes. How does it affect the behavior? Again, we wanted to evaluate that.
And so these are known a priori, there's some taxonomy of these?
Well, it depends on. For this, we use t-test or different type of methods to identify. If we know different groups, so we know this is T cell, this is B cell, this is NK cell, we ask the question which genes are uniquely expressed across this group that is not expressed in other groups. And also we ask the question, is it consistently expressed within that group, or there is high variation within that group as well. So
when you remove the signal from the cell classification, are you not losing a lot of information? Because cells are sometimes very specific.
yes, so I do agree. So for better or worse, a lot of tissue and cell type specific genes are known to be receptor genes. However, what we are keeping is the upstream ones, which are receptors, because their behavior is consistent. What we are removing from them is the upstream signaling element, so things that are right below membrane till the transcription factors. So we are removing factors in the signaling pathways, but receptors themselves shows consistent behavior. CD4 shows consistent expression. CD8 shows consistent expression. What is downstream of it, what is sensed, is the one that shows differential behavior. So we're keeping those cell type specific components, but we're removing the connection between those till the changes that they make. And there are some additional constraints, for example, on solutions. Some use least squares and remove non-negative elements afterwards. Some put that as part of the problem. Same with sum to one, and there's additional constraint called similar cell quantity, which we're going to look at. And then we looked at different values, different combination of these factors over 7 different data sets. And ask.
Sorry, can we just understand this a little bit more? No. Your next slide.
This one?
Yeah.
Sure.
Oh, sorry. Are you about? It looked like you were going to jump through this one, but so the features are.
Features are genes.
Okay.
So if we get this tiny skinny matrix, genes, samples, So genes, samples, number of cell types. Those are the three factors that I'm looking at. Genes,
And number of references
samples,
is what?
number of references.
What is number of references?
Number of cell types. How many cell types do I have? Those are reference profiles that I want to decompose to do those reference profiles.
Okay.
Then we had multiple lessons. Lesson number 1.
True, or are some of them synthetic? Are these mixtures real mixtures?
Majority of them are actual in vitro mixtures where they mix these in known proportions.
Which one of these is not in vitro?
These two.
In vitro? you
These
said
are in vivo
in
mixtures.
vivo.
The rest of them are in vitro, and the behavior are significantly different. That's we are going to look at and see. So number 1, which we observe, which I found.
So the features, there are a lot of overlapping genes as well, right? Or they are all unique genes?
Which
So
ones?
the features are
Each one of them I look at individually,
the
right?
number of features, is that number of genes?
Same genes between references and samples, identical number of genes that I'm looking at. So I'm looking at matrices that G and M, they look same length, different width.
But they could be overlapping, right? Similar genes across different.
Sure. But I'm not comparing across the data sets. But yes, they might be same genes. Like here, they use the same technology, so they looked at same probes, so they're identical set of features.
So silly question, but everyone always says humans have 30,000 genes.
So these are probes. Probes are gene fractions. So what I do is that I want to find this gene. I randomly pick this region, this region, this region, and put probes to look at them. And then I have more features than genes. That's the 50,000 that we are looking at. Some of them are 20K. Those 20K ones are almost the number of genes. So if you look at number of genes that we have in human, it's 20K-ish that is measured. So it depends on what are your features. Are they genes? Are they probes? Are they... so each gene might be also multiple mRNAs. So number of mRNAs are larger than genes. So it might be different things that you are measuring.
And do you have independent replicas of any of these data sets?
These are, yes, number of samples are replicas of what you have, right? So
No,
number of samples are...
I'm saying like breast blood was collected by, some group at MIT.
Okay, so these guys are...
Did that group collect that, a week later? So you have a
These two are.
independent-
These two are.
Okay.
These two, I explicitly have them because their references are from somewhere else, and then they use those references to actually convolve an in vivo mixture, which they have been in different condition. There have been like a lot of changes in between, and the behavior for the convolution of this is significantly different than the rest of them, as we're going to see. So,
Just to poke at this one more time. So it seems like there's a lot of things going on here besides the nice setup. There's a lot of different things that are mixing here, and now you're going to look at these with all this mixed.
no, I'm going to look at the properties of them. So the first one, I'm going to say that purge behaves differently than breast and blood, for example, and why that behaves differently. We are going to go over differences and say why those differences.
But why would you expect it to behave the same?
Because, well, if L2 norm was, so people usually use L2 norm. If it was a good norm, which with modification is, it should have behaved well in all cases. What we are saying is that if references come from somewhere else, you should not use L2 norm without regularizer, without pre-processing. So we are looking at everything that people use. Those reference profiles that we look at are de facto reference benchmarks that people use. So we use the same benchmarks. We used every components that they used mix and matched and said which one of these components that they mix and match is the reason that they behave better than the rest of them.
But are you attempting to do any type of noise characterization of the underlying process?
No. What we are trying to say is that these are the data sets that we want to do the convolution. These are loss functions, these are regularizers, these are pre-processing. If we do one by one, mix and match these, which one of them is the source of better response. So they say SVR is better than this one. Is it because it used epsilon insensitive, or maybe if we use L2 with regularizer, it performs even better than SVR? So we wanted to say there are so many ad hoc choices in literature that each one of them there's a method. A combination of these, some of them are already a method, some of them are like novel combinations, but people were using a random mixed match of these in order to solve the convolution. And the problem was why one deconvolution problem works better in some data set than the rest of them. Can we figure out what are the winning factors for one method versus the other methods? So number one thing that we figured, find out is that robust measures behave similarly. So L1, L Huber, and epsilon S insensitive loss behave similarly if you do cross-validation to find epsilon and m, which is interesting. So what it says is that you don't really need to use SVR, which uses epsilon insensitive. You could have used L1 to begin with and got the same result. To interpret this result, this y-axis is error, and the x-axis have blocks of 4, L2, L1, Huber, epsilon insensitive. And implicit and explicit tells if we are doing the constraint as part of the program, or we are solving it, then enforcing those properties. So L2 implicit means that we used linearless squares, then we removed every non-negative elements, then we column normalize the factors.
You want big or small on this plot?
Small means less error, so better. Large means more error, bad. So in breast and blood, where references come from the same place, having a more sensitive L actually performs better, as we expected. But what we didn't expect, in a sense, is that these almost behave identical to each other. L1, L, epsilon insensitive, and Huber. So you may as well use the one that is non-parametric, and you don't have to deal with fine-tuning it. And in other words, performance can be measured only by looking at L2 and L1. So
these bars are these medians over some cross-validation, or
overall sim. It's mean absolute difference, so it's mean value across different samples. And it is consistent with RMSE values. So we looked at two separate measures. They were consistent with each other, so we used mean absolute difference, which is easier to interpret. So it says on average we have 6% error in estimating fractions. That's what it means. Then
you didn't do any subset analysis to make sure that there's,
a group of them might have more error than the rest of them.
well, I mean, you could, if you've got 50,000 things and you're looking for like 3 or something like that, so you could dramatically subsample the number of rows that you have.
We are looking at the others, right? We are looking at the ones that was tens. We are looking mean over samples, not mean over genes. We are looking at fractions of each sample, fraction of cell types in samples.
I think David's point was, how do you know that your markers are correct?
We still haven't looked at it. Further, we are going to look at it.
For markers in that experiment?
Here we are looking at markers.
In the previous experiment, what?
Everything is there.
Everything.
Everything is there.
All the genes are equally considered.
Yeah.
And what was the deconvolution method?
We looked at this many deconvolution methods, which are different loss functions with implicit or explicit enforcement of different constraints. So it was either...
You were doing NMF or what were you doing?
Here you are doing NMF, which is, explicit, implicit. So non-negativity is explicitly enforced, sum to 1 is not enforced with L2 norm, so that's non-negatively squares. NMF.
What do you mean when you say you solved a non-negative matrix problem then?
It's what it means is that I want to do GC factorization. I put a constraint on C matrix to be all positive.
But you then can't solve that objective function analytically.
If you have G, you can solve it, right?
Oh, so you're fixing G?
Yeah.
Okay.
Given the reference profile, you can solve for C.
Okay, sure.
So the second one was to answer your question, how does choosing subsets of genes affect the behavior? We look at two properties. One is if you use t-test, which is the one to find markers, in order to find what are the genes that are more cell type specific. Does it behave better or worse? In one data set, rat brain, that had the least error originally, we observed so here we say how much the error changes. So 0 means get the same result, positive means the error is reduced, negative means error has been increased. So negative is bad, positive is good, 0 is no change. So what we observe is that we have two data sets that we overfit over. So the breast and blood, we already showed that L2 is very good. It's kind of overfitting because references come from same place. So the large values are the ones that actually drive this the convolution, and those are the ones that we are sensitive to. We don't want to remove those high values. Same with this rat brain, references come from same one. The parts where the references come from somewhere else significantly enhance the result when you look at the subsets of genes that are cell type specific. So what we are saying is that if your references come from somewhere else, which as Dr. Gama was mentioning, it has different microenvironment, different signaling factors, there's a lot of change that happen in the reference profiles. And in that case, you need to use some pre-filtering of genes. And here we introduced one additional factor for filtering. We said we want mixture to be represented as a complex combination of a bunch of genes. If the minimum of those genes is larger than the mixture, there is no way you can match that. And if the maximum is smaller than mixture, there is no complex combination that can match that. If you remove those genes that are violating gene, in almost all cases, except that one that I told you is highly overfitting, in almost all cases you get better results than what you were getting before. Finally, we observed that L2 regularization almost always enhances our results, especially in those two cases where references come from different places. To conclude, what we observe is that if you use the right combination of regularizer, which is L2, and you use L2 norm for loss, and you do pre-processing for finding genes that are actually cell type specific and are low variation, you get consistently good results across different data sets regardless of the fact that these two are coming from different places, the reference. So originally the gray one was the original performance. With tweaking with the elements of the convolution, you get to the lower ones. What it shows is that L2 almost performs better in majority of cases, and in almost all cases you can get it around 5% error. So finally, I have almost 5 minutes, 10 minutes? Quickly, okay. So this one I wrap it up really fast. So the idea is we have these networks that are a mishmash of all things that can happen everywhere, anywhere, whatever. So we looked at these protein-protein interaction networks. In reality, what happens is that you put these proteins in yeast and look at if they can interact with each other. That's assuming that these proteins are expressed. That is assuming they're in close proximity of each other. That is assuming that that context allows whatever condition that is needed for these two proteins to interact with each other, which either one of them might not happen in reality. So the question is, can we use additional source of information to prune this big network to the interactions that are actually happening in a given context? The answer is, well, yeah, we can. And the way we do it is that we start with a network of transcriptional activity. So red ones are genes or proteins that are not expressed, green ones are the ones that are expressed. Then what we do is that we say we have a hypothesis, a gene is functionally, so this is transcriptional activity. What we want to estimate is functional activity. A gene or a protein is functionally active, it's within a functionally active context. So these are two protein complexes where one of them is active, one of them is not, and there are some mismatches in between. Two signaling pathways, one of them active, one is not, where there is some mismatches. What we want is to use a diffusion process to correct for those mismatches. And then this is only at the node level. We need to project it back to edge level, and then we prune the edges that seem to be not used in that context. So in order to solve the diffusion process, we use a comics program where Z is the transcriptional activity, or those red-green stages that we are looking at. And then we want to make minimum number of changes to the order. So we say this is almost correct, but it's not absolutely correct. So we want to do minimum number of changes, but we want to also behave as switch-like. We say if you're absolutely sure you need to change it, change it. If not, leave it unchanged. That's why we use L1 norm there instead of L2. We want to minimize the number of changes that we make, and then we want to also propagate those changes such that neighboring vertices have similar functional activity scores. So then obviously alpha balances the contribution of these two factors, where we cross-validate to find the optimal alpha. And then we project them to the network using a simple approach that says this is the edge weight of this one, 0.5, whatever. This is the original edge weight. We multiply it by functional activity of the two endpoints. So functional activity is computed as the solution of this program, and then what we do is that we reweight original edge weights by the endpoints of that functional activity of its endpoints. And we use GTEx data set, which has a bunch of different tissues, a different number of samples. We constructed tissue-specific networks here, and some of them are cell type, if I'm remembering correctly. And then looked at how does it behave. This one we don't care, we don't have time for it. Just what it says is that the expression profiles can predict tissue-specific genes. So if we compute tissue-specific genes, what we observe is very relevant to the tissue that we are looking at. Then the second step that we validate is we have these known pathways. If we have known tissue-specific edges in known pathways, can we predict those using the tissue-specific networks that we have? The question is, we can, and this is differences in AUC. So the baseline, the random baseline is 0.5. We say how much better than 0.5 do we get on average across all tissues. And these three are ActPro, which is our method. These two are the other two methods, which says the other two methods almost act randomly. So they don't predict any tissue-specific edges on average, and the method that we have performs better. To be honest, the problem is if you look at these different tissues, some of them perform really well in terms of AUC, some of them perform really bad, and some of them perform worse than random. And that's why, on average, these are almost around 0.5. To give you one example of how do they behave, in one of the examples that it behaves better than the others, the AUC goes for these three methods that are different values of alpha and ActPro, it goes up to 0.8-ish, and the other two methods are 0.7-ish. So far so good. We can predict tissue-specific edges in known pathways. The other one is, if we use these networks, can we predict disease-specific genes in tissue-specific disorders? For example, if we use brain-specific network, can we predict Alzheimer's disease-related genes better than using the global network or networks created by other methods? Same for a bunch of other disorders that are tissue-specific in the behavior. And the final answer is yes. With ActPro, we get better p-values in predicting known tissue-specific genes compared to global network and networks coming from other methods. And finally, we said, okay, we know that we can predict disease-related genes. Can we identify disease-related pathways? And the idea was we start from some known disease-related genes. Can we use tissue-specific networks, here brain-specific network, to construct pathways that are involved in brain-specific pathologies? The answer is yes. We validated all interactions and the factors that are novel. So red ones we already know. The rest of them we found. And what you found as additional connecting nodes, we found that there are also evidence for all of them in literature that they should be related to Alzheimer. I really skimmed over this last one. We did this. We went through 3 projects. One was, can we identify cell types using tissue-specific, so using single-cell data set. The other was, given a mixture of cell types, can we separate them from each other? And finally, if we have cell type-specific transcriptome, can we build cell type-specific networks? And there are a bunch of things that I want to do in future. Comparing networks, tissue-specific networks, combining single cell with deconvolution, and increasing the number of cells. So far, we can deconvolve in order of tens of reference profiles. Can we develop some techniques that we can increase that dimension significantly, because we are identifying more cell types using single cells, so we need to increase that dimension somehow. And I want to thank everyone that helped me during past 7 years in different specs, their advisors and people that allowed me to go visit their lab, and Dr. Gleich that was really helpful in all the conversations we had in different papers, in multiple different specs. Thank you very much. You were one of the smartest people I worked with, that I had the pleasure of working with. It was a pleasure working with you. It's an honest opinion. And other people, co-authors that helped me in different papers. And finally, a bunch of papers I had to cite, of course. And questions.